we certainly want to have enough time for the last talk of the conference, and this one is from someone who's traveled across country to be here, and that's Dr. Tracy Young-Pearse. And Tracy, Dr. Young-Pearse, got her original degree at Skidmore College and then got a PhD in genetics at Harvard. And she followed that by doing a postdoctoral fellowship in the laboratory of Dennis Selkoe, a very famous and influential paradigm-shifting individual in Alzheimer's disease. And this was also at Harvard at the Center for Neurological Diseases at the Brigham and Women's Hospital. Now, what Dr. Young-Pearse is doing is using more molecular approaches rather than what we just heard of as a sort of rather innovative combination of the imaging tools and psychological testing as well as drugs. Dr. Young-Pearse is really studying more the molecular basis, and she's trying to determine the function of different genes in the pathogenesis of Alzheimer's disease, as well as neuronal development and other neuronal disorders. And so she's going to talk to us about her really innovative studies on stem cells and how that relates to Alzheimer's disease, neurodegeneration, and then hopefully some treatments for the disease. Thank you very much.
Thank you so much for that kind introduction, and also thank you so much, Andrea, and to the other organizers for inviting me here today. It's really been a wonderful experience being here today and hearing all of the great talks, and seeing the panel today was really quite moving and has re-inspired me to get back to the lab to do some more research on this important topic. So, as you heard, today I'm going to tell you about the studies of my lab over the past 3 to 4 years since I started my lab, in trying to understand the molecular basis of Alzheimer's disease specifically, and using stem cells. And so the ability to generate adult stem cells from adult patients is a relatively new technique, which I'll go into further in the following slides. But previously, there are many other, and currently there are many other ways to study mechanisms of disease, and you've heard about all of these today and more. So Dr. Green, just a little while ago, talked to you about different animal models that we can use where we can introduce human disease mutated genes into a rodent, and we can see pathology and we can see behavioral defects. And we can see this in an intact brain, in an intact system, where we can then try to rescue those effects by testing novel therapeutic interventions. And he talked a little bit about his work destroying microglia in that case. However, the one problem with animal models, and I use animal models too, quite a bit, the one problem with animal models, of course, and Kim mentioned this as well, is that there could be very important differences between the mouse physiology and the human physiology. And so we really want to complement animal models with studies in human cells. And we just heard from Mike about some imaging studies in living patients where you can look at different pathways and different pathologies as well to look in the living patient at what could be going on as the disease progresses. We also have, of course, post-mortem analyses that can tell us a lot about the pathology and the types of proteins that are accumulating abnormally in Alzheimer's disease, for example. But the one drawback for these studies are that they're not that manipulatable. So we can't mess around with gene expression in humans. We can't, we can give them drugs to test the drugs, but we can't screen through many drugs that may be dangerous and have important side effects. And so this ability for us now to generate human neurons in a dish from induced pluripotent stem cells is really going to, I believe, revolutionize the way we look at, we test for novel therapeutics. And so certainly there are drawbacks to using stem cells to model disease as well, but I think that there are very important complementary techniques that we now have in our arsenal to try to understand the disease process and also to test therapeutics. So one important feature of neurodegenerative diseases is that in different neurodegenerative diseases, of course, different brain regions are attacked. And so in many different diseases, so for narcolepsy, all right, that's the one. In narcolepsy, for example, you have a very small number of cells that are degenerating over time, and this can cause problems with sleep and wakefulness. Whereas in Alzheimer's disease, we have degeneration of the there we go, of the cortex, cerebral cortex, and hippocampus, the forebrain, while other regions of the nervous system are spared. And so, as a molecular biologist, I really would love to be able to study what is different about these cells. And to do this, I really want human cells of these different types of brain regions, different fates. And until iPS cell technology became available, this is really almost impossible to obtain. So from a living patient, to get a brain biopsy from different regions of the brain and compare those cells was virtually unheard of. However, in 2007, Yamanaka, with his Nobel Prize-winning discovery, identified a method to take adult skin cells, and now we can do this with blood or hair or any cell that's dividing, and transform those skin cells into stem cells by introducing just 4 transcription factors. By doing this, we get these patient-specific iPS cells. So we have a patient with a very specific, perhaps genetic abnormality that we have identified or that we haven't yet identified. And we now have cells in a dish that have the exact same genome as the patient that we're studying, that we're interested in trying to understand their disease. Now, there are 2 very important features of stem cells. One is that they're immortalized, which means that they will continue to divide in culture for eternity, in theory. And so we have an unlimited supply of these cells. The second important feature of stem cells is that they're pluripotent, which means that given the right cues, a researcher is able to direct these cells to become any type of cell in the body. And if we're able to do this, in the case of Alzheimer's disease, we want to look at things like neurons, astrocytes, microglia, and we want to try to then try to use these cells and probe the mechanisms of disease, probe what the functions of these genes are and what's going wrong in the disease state. We also want to be able to test novel therapeutic interventions in the cell types of interest. And finally, in some cases, for some diseases, cell replacement therapy may be an option. And having cell lines, neural cells that have the same genotype, the same genome as the patient that they're introduced to would be really an important new development. So here in the middle here is a picture of a colony of stem cells. There are tens of thousands of cells in this dark circle. And what we do as researchers is we try to use our knowledge of normal developmental processes to push these cells to become different types of cells in the body. So we look at how in the embryonic human brain, the different neurons are generated and what factors are important for generating different types of neurons. We try to recapitulate that environment in a dish to try to generate these different types of neurons. So over the past 3 to 4 years, my lab and many others have optimized methods to try to direct these stem cells to become cells of the brain, so neurons and glia. And I won't go into the details of how we do this, but there are basically different types of factors we add to the media over time that changes. And we also change their culturing conditions and the types of vessels that we culture the cells in. And so you can see over the course of 60 days, over the course of 2 months, we're able to transform these pluripotent stem cells into cells that look very much like neurons. And not only do we see a change in the morphology or the shape of the cells over differentiation time, but we also can mark the different cell types with using antibodies. So here we've marked the pluripotent stem cells in red, and you can see over time this red signal goes away. We've marked the neurons in green, and you can see the onset of this green signal. And then by day 60, after 2 months, we have most of the cells having neuronal fates. So we're really excited now because over the past 3 years, after developing these methods, we now have a virtually unlimited supply of living human neurons from our patients in a dish. And now we can start to try to understand what's different about these cells compared to cells from healthy control patients. And we can also test, as I said, novel therapeutic interventions. And everyone in this room, we've heard a lot about Alzheimer's disease today, and you know that some parts of the nervous system are affected and other parts aren't. So in advanced stages of the disease, you have various problems with cognition, memory, thought, awareness, attention, and consciousness, but you don't have problems with things like movement and balance necessarily in most cases of Alzheimer's disease. So today in the panel, I was struck when Lucy was talking about having trouble reading and maintaining attention in conversations, but then she said, but she has no problem running up and down the stairs. So this is an image that Kim showed also. That's a very popular image because it really highlights the degeneration and the cell loss that occurs in the cerebral cortex. But here, if you look at the cerebellum, which is important for balance and coordination, you see that this is relatively intact. Now, prior to this overt cell loss in the cerebral cortex, we see the accumulation of the pathological hallmarks that you've heard about today, the plaques and the tangles. And we don't, in most cases of Alzheimer's disease, we don't see this accumulation in regions of this, like the cerebellum and the spinal cord. Just to remind you, these, because I'm going to talk a little bit more about tau and A-beta, the tangles, the intracellular tangles that appear are composed primarily of hyperphosphorylated tau. And these brown extracellular deposits of protein consist primarily of a peptide called A-beta. And very early up today in the morning, you heard that A-beta is generated by sequential cleavage of APP in 2 places, and that you can have different regions, different areas where these scissors will cut the protein, and you can get different lengths of A-beta. And we believe that this longer form of A-beta is more prone to aggregation and more toxic to the cells. So just to keep in mind that we all have APP in all of the cells of our body, and we're all making A-beta of both forms. But we believe that in some cases of Alzheimer's disease, we have an increase in this longer form that leads to disease. And we believe this is especially, the evidence is particularly good for this early onset form of Alzheimer's disease that Ken Kosick talked about earlier today. So Ken told you about this dominantly inherited, fully penetrant mutation in presenilin 1 in the Colombian kindred. And that gene, presenilin 1, encodes the active site of this protein here that cleaves APP to make A-beta. Now, in Boston, we had a family who also had a very early onset form of Alzheimer's disease. And this family, instead of having a mutation in the enzyme that cleaves APP, actually has a single amino acid change in the APP gene itself. And so this valine that's in the membrane of this large protein APP is switched to an isoleucine in this family. And if, again, just like in Ken's family, anyone who has that mutation is destined to develop Alzheimer's disease very early in a very severe form of the disease. And children of those subjects have a 50-50 chance of inheriting that allele and developing the disease as well. So today, to highlight the pros and cons of modeling neurodegenerative disease with iPS cells, I'm going to tell you about our work with these familial Alzheimer's disease lines that we generated from these patients that have this APP mutation. Just as an aside, I'll mention to you that we're able to actually go into the genome of these cells and fix this mutation in a dish. So we can go in with something called CRISPR-Cas or with TALENs, and we can correct the isoleucine in these cells to a valine. And so in that way, we can compare both cells that have the mutations and cells that don't have the mutation, but the rest of the genome is entirely the same between those two, the control versus the familial Alzheimer's disease cells. So this is an overview of the amyloid hypothesis of Alzheimer's disease pathogenesis. And as I said, this evidence for this is very strong for familial Alzheimer's disease, where we have missense mutations, single amino acid changes in APP or the enzyme that cleaves APP to generate A-beta. There are hundreds of mutations around the world in these genes that cause early onset Alzheimer's disease. And all of them that have been tested have been shown to either increase the overall levels of A-beta or increase this A-beta 42 species relative to the A-beta 40 species. So we believe the ratio of A-beta 42 to 40 is a very important pathological hallmark of at least the early onset form of disease. We believe that over time, this A-beta accumulates and has effects on synapses that lead to mild memory loss, but that as time progresses even further, we have changes in inflammation, where we see microglial and astrocytic activation, as well as these changes in tau that lead to tangles, so increases in tau levels and phospho-tau levels. And ultimately, these leads to neuronal cell loss and dementia. So we wanted to investigate whether we were able to recapitulate some or all of these aspects of what we know what we know happens in vivo in the patient's brains, and see if we can recapitulate this in a dish. So I told you about how we've optimized methods to generate brain cells in a dish. And when we look at different markers of different proteins in neurons between control neurons in familial Alzheimer's disease neurons, we really see no differences in the ability of the stem cells to make neurons, which isn't surprising. These patients don't have very different developmental processes compared to, as far as we know, they have no developmental differences between themselves and non-carriers of the mutation. We can also look at we can look at more things in these cells as well. We can look at RNA expression of a number of different neuronal markers, and we see no differences, again, in many of the genes expressed in FAD versus control neurons. So they're very similar neurons that we get in a dish. However, if we look in the media of those cells at the A beta that's secreted, we see exactly what we had predicted that we would see based on what we know from the patients, which is we see an increase in this pathological 42-40 ratio. So with FAD mutation, we have more secretion of A beta 42, with no real change, no statistically significant change in the secretion of A beta 40. And also interestingly, we didn't predict this, but we see also that an increase in a very short form of A beta 38. And here we're just showing that we can also add an inhibitor of gamma secretase, an inhibitor of that enzyme that cleaves APP, and we're able to in the dish, we're able to completely block secretion of A beta of all species. So one of the powerful aspects of stem cells is that you can look longitudinally at them over time. And here we're looking over differentiation time from day 9 to day 100, so these can be quite long experiments. And over differentiation time, we see as cells become more and more mature neuronally, we see more and more secretion of all 3 A beta species. But at all time points when these cells have a neuronal phenotype, we see an increase in the amount of A beta 42 secreted by the FAD cells. Here I also wanted to put up this graph because, again, everything we're looking at here is all the A beta that's secreted in the media. But in this graph you can see we can also look at the A beta that's present inside the cells, and it's really at quite a low level relative to the A beta that's secreted from the cells. So we think that the A beta can have effects that are non-autonomous. So the A beta can get secreted from neurons and have effects on neighboring cells. So we've shown that this mutation affects the precise site of this gamma secretase cleavage such that we get longer species. One thing that we weren't expecting to see, but that we observed, was that we also have effects on this other cleavage event in APP. So A beta is generated by cleavage first by beta secretase, then by gamma secretase. And we see that with this mutation at the gamma secretase site, we also see an increase in the cleavage at the beta site. And we see this consistently over differentiation time, that the FAD mutation increases the beta secretase cleavage of APP. And one interesting feature of this is that beta secretase cleavage happens in specific subcellular compartments. And so we thought perhaps there was an increase in beta secretase cleavage because with this APP mutation, perhaps APP had an altered localization within the cells. And indeed, we see that this is the case. So in green is expression of APP within individual neurons, and red is marking this subcellular compartment that is important for beta secretase cleavage. And we can see that we have a change in where the APP is in the cell. So we know a little bit more about what could be going on in these subjects that leads to Alzheimer's disease, but we really wanted to look beyond this really what I call proximal phenotype to the APP mutation. So we see a mutation in APP, we see effect on cleavage of APP, so we're able to recapitulate some of the aspects of the disease process, but we also wanted to look at downstream effects. So one of the questions when I started this project was, are we going to be able to recapitulate aspects of Alzheimer's disease in a dish in a very short amount of time, relatively short amount of time, right? So in patients, this disease, even with the mutation, takes decades to show symptomology. And here we're asking the cells in maybe just 30 days or 60 days or 100 days to show molecular changes. And we weren't sure if we would see these molecular changes, but I'm going to show you a little bit of data that suggests that we see some molecular changes that may reflect very early events in the Alzheimer's disease process. So one of the first things we looked at was tau, because of course tau is the other pathological hallmark of Alzheimer's disease. So hyperphosphorylation of tau. And at day 35, at just 1 month after differentiation, we see no change in the levels of phosphorylation of tau, but we did see this interesting increase in the overall levels of tau. By day 100, this increase in the overall levels of tau persisted, and now at this point, we were able to see an increase in phospho-tau even when we normalized to this increase in total tau. And this is something that we're trying to better understand now is, what does it mean to have an increase in total tau, and could this be a necessary predecessor to an increase in phosphorylation of tau? I mean, I don't have an answer for that. We're trying to understand that now. But separately, by seeing an effect on tau here, it gave us another phenotypic readout for testing therapeutics to see if we lower A beta, can we alter this tau phenotype as well? So is this tau phenotype due to the altered A beta cleavage that we saw, that I showed you earlier? So here we used antibodies, a monoclonal and a polyclonal antibody, to try to neutralize the effect of A beta in the dish. And so here I'm just showing that the antibody, one of the antibodies binds efficiently to A beta, and that all of the A beta seems to be bound to these antibodies at the concentrations we're using, because if we pull the antibody A beta complexes out of the media and then we measure the amount of A beta that's left in the media, the free A beta, it's virtually undetectable. With these treatments in these cells for about 14 days of antibody treatment, we see that we're able to significantly reduce the overall levels of tau that I showed you before. So here again, this is with the monoclonal antibody and with the polyclonal antibody, looking at tau levels by ELISA and by Western blot, 2 different methodologies. And again, in the control groups, we see that with FAD mutation, we see an increase in overall tau protein levels, but with the antibody, we're able to significantly reduce these tau levels to near normal levels. So this was important for us because it showed us that the tau phenotype that we're seeing recapitulated in the dish is due, at least in part, to a change in A beta levels with the mutation. So in addition to looking at changes in tau, we also were wondering if our iPS-derived brain cells would also recapitulate this aspect of increased astrocyte activation. And I'm actually that's kind of a lie. We didn't really think that there would be any changes in astrocyte activation. We weren't sure, we didn't think that this would happen this quickly, and by accident we kind of realized that there was a change in astrocyte activation. So just to remind you of our overall schema for generating neurons that I showed you earlier, this is over the course of 100 days here, different media changes, different vessel changes. And what I didn't mention before was that around day 40, we start to get the emergence of endogenous astrocytes from these cells. So in normal brain development, the same progenitors that give rise to neurons also give rise to astrocytes. And indeed, in a later time, then neurons are generated in the embryo, and indeed this is recapitulated in the dish, where by day 40 there are still some remaining progenitor cells around that then divide and give rise to astrocytes. And we can see this not only at the immunocytochemical level, but also at the RNA level, where we see increases in astrocyte markers. And just as an aside, we believe that this emergence of endogenous astrocytes is very critical to maturing the neurons in terms of spontaneous electrical activity. We can see that there's a correlation between the increase in astrocytes and the increase in mature neuronal markers that we see. And in fact, if we co-culture exogenous astrocytes with our early neurons, we're able to mature them faster. So when we looked at day 100, which is a time point when there are these endogenous astrocytes, we were looking at a number of different genes just to see how these neurons might be different between the familial Alzheimer's disease neurons and the control neurons. We saw no strong changes in general neuronal markers, and we saw no change in one of the astrocyte markers, S100 beta, but we did see a very strong increase with FAD mutation in the expression of GFAP at the RNA level. So GFAP is a marker of activated astrocytes. And not only did we see this this is what we were first surprised by. We said, geez, if it's up at the RNA level, is this real? Is it also happening at the protein level? And indeed, by Western blot, quantified here, and also by immunocytochemistry, we can see that these astrocytes in the FAD cultures appear to be more activated in terms of expressing higher levels of GFAP and also secreting more of the cytokine IL-6, which I'm not showing here. So we wanted to test whether there was something about if the mutation could induce this effect in a non-autonomous way. So was the A beta coming from the neurons activating the astrocytes, or was it that the mutation within the astrocytes was causing them to be more activated? And so here we took wild-type human astrocytes, so that don't have the mutation, and we co-cultured them with control neurons or with FAD neurons, And now these wild-type astrocytes were exposed to the FAD environment or the control environment, and we saw that we were able to see, we did see this increase in activation of these wild-type astrocytes, suggesting that something was being secreted by the FAD neurons, and of course, we jumped to A-beta, because that's what we believe is primarily changed with this mutation. And so here, we take conditioned media from FAD cells, and then we either immunodeplete A-beta, which means we remove the A-beta from these cells, or we leave the A-beta in there, and we can see that, again, we see this activation of astrocytes with FAD conditioned media now, and we're able to rescue this effect by immunodepleting the A-beta from that media, suggesting that A-beta, not only can this altered A-beta that's resulting from the APP mutation, not only increases the level of tau and ultimately phosphotau, but also acts to activate astrocytes. So I hope I've, at least in part, convinced you that we're able to recapitulate some of the early events, possible early events, in familial Alzheimer's disease progression. And we really want to use this system to try to test novel therapeutic interventions, but also to test new genes that are coming out in the GWAS studies that you heard about earlier today, and try to understand how these fit into this Alzheimer's disease pathway by manipulating them in these cells in a dish. In addition, we're also very interested in studying this idea of differential vulnerability that I mentioned at the beginning of my talk. So as you know, and as I mentioned, some cells, such as those of the forebrain, the hippocampus, and the cortex, are highly vulnerable in the disease, both to pathological hallmarks, but also to symptomology and to degeneration. So what we want to do now is try to take our iPS cells and direct them to be, everything I've showed you so far has been directing these to these cells that are vulnerable in the disease, but we'd also like to direct them to other neuronal fates, so neuronal fates that aren't usually affected in Alzheimer's disease, to try to understand why are some cells in the same patient where A-beta and tau are present throughout the central nervous system, why are some neurons dying and other neurons are protected? And if we can understand what's protecting different brain regions, maybe that would give us some clues as to how we could try to protect those vulnerable cells. So there are many labs around the world trying to make different types of neurons, and we're one of them, and we've used the information that has been published by a number of different labs to direct these neural progenitor cells to become neurons of a more caudal fate. So these neurons, when we add retinoic acid and sonic hedgehog, which are similar factors that are important in normal development to push cells to be neurons of that fate, we can push these cells to be different types of neurons, and we see changes in gene expression in these neurons. So we see no change in general neuronal markers at the RNA level or at the protein level, but we do see, when we push cells to be more caudal, a change in gene expression. And we believe that even though we have very heterogeneous cultures, we have different subsets of neuronal types in this cortical versus caudal cell fates. And interestingly, what we saw when we looked at A-beta secretion from these neurons was that when we pushed the cells to become these more vulnerable fates, we saw overall an increase in A-beta levels, which was surprising. We also saw a change in this A-beta 42 to 40 ratio. So if we take this data, where we are looking at A-beta 40, 42, and 38, and we look at the ratio 42 to 40, and we normalize the cortical neuron secretion pattern to 1, you can see that in both control lines and in FAD lines, a decrease in this A-beta 42 to 40 ratio, which was somewhat surprising, but pretty interesting to us that the precise site of gamma secretase cleavage may be subtly altered in different neuronal populations, and we're trying to better understand that now. But even more interesting to us was not only did we see a change in APP in the site of gamma secretase cleavage, but we also saw a change in that tau phenotype that I described earlier. So in forebrain neurons and cortical neurons, this is what I showed you before, that we see an increase with FAD mutation in total tau levels and phosphotau levels. But when we direct these neurons to be a different fate, a different set of fates, we no longer see this effect, and in fact, we see a slight decrease. And so we're trying now to understand further what this means, but we're hoping that this iPS cell technology is going to allow us to further probe why some neuronal subtypes are vulnerable in the disease at a molecular level, and why some are protected. Now, as I alluded to a few times as I was speaking about this, when we direct it to these different fates, we have dozens of different neuronal subtypes within those dishes. So even though these are cortical, within the cortex, there are upper layer neurons and lower layer neurons, there are inhibitory neurons and excitatory neurons. There are many different kinds, and similarly so in these caudally directed cells. And so even though we see changes in A-beta cleavage and tau levels, it's likely that there are particular neuronal fates within these heterogeneous cultures that are contributing more or less to the phenotypes that we're seeing. And so over the past few years, we've wanted to look more specifically at very specific neuronal subtypes. And to do this, we've developed an assay to measure A-beta secretion from individual single cells. So this was done in collaboration with the Love Lab at MIT. Chris has developed this technology to study cytokine secretion and antibody secretion from B cells and T cells. But we thought perhaps we'd be able to optimize this for detection of A-beta from iPS-derived neurons. So this technology involves having a slide of tens of thousands, 86,000 little tiny nanowells that each can house a single cell, or 0, 1, or 2 cells. We then plate our iPS-derived neurons at a density that favors a single cell per well, and we seal each well off from its neighbors with a glass slide that contains antibodies that can bind to A-beta, that can capture the A-beta. So we leave the cells in these sealed nanowells for about 4 hours. Because there's only about 100 picoliters in each well, we have an exquisite sensitivity where these living cells are secreting their A-beta or not. The A-beta is being captured by these antibodies that are bound to the glass slide. And then after 4 hours, we peel off the glass slide, and like a sandwich ELISA, we can detect the amount of A-beta that's secreted with a fluorescence conjugated secondary antibody. And we're able to see, this is just one block from this 86,000 set of wells, some cells are secreting high levels of A-beta and very low levels of soluble APPS alpha. Oh, sorry, I didn't mention we're co-detecting 2 different secretion products of APP. This secretion product, if you have secretion of this, it lowers the secretion of A-beta. And we can see some cells that secrete both and some cells that secrete none. And what we can do is then say, what's different about this cell that's secreting high levels of A-beta compared to this cell? We can then go back to the cells that are living in the nanowells and either immunostain those cells for different neuronal markers, or else retrieve those cells from the well and then do gene expression profiling. So it's still early days for this technique, but we're now able to efficiently, I shouldn't say efficiently, it's a low efficiency, but for about, in a population of iPS cells, we can detect A-beta from about 5% of the cells that are present for a given 4-hour period. And here I'm showing histograms of A-beta secretion from 1 or more cells that our algorithms determine are positive for secretion. And we're comparing these to the blue line, where we can compare to wells that have no cells, so we can see what our specificity is of our assay, and we have good separation there. So then of these positive cells, we can look at the distribution of A-beta. Let's focus here. The distribution of A-beta signals that we see when we have only A-beta secreted, or that we can detect, versus when we can detect both APPS alpha and A-beta. And here, what's powerful about this is that we can co-detect these 2 analytes from the same single cells and plot them like this, where we can see cells that secreted very high levels of A-beta and very low levels of APPS alpha, and try to understand what the fate of those cells is. And I don't have an answer for you today, but we're getting there. And this is just some of the evidence that we're getting there. We can look at cells that are secreting high levels of both. This is just a single nanowell with a single cell, secreting high levels of both, and we can immunostain for different markers to try to figure out what subtype of cell these are. And again, this is early days, but using a single cell nanostring protocol, we can amplify the cDNA. We can look at the RNA expression pattern of a number of different genes from a single cell. So to summarize what I've told you today, we've generated new stem cell lines from patients with an early onset form of Alzheimer's disease. In all neuronal fates tested, this mutation leads to an increase in total A-beta and A-beta 38 and A-beta 42. A-beta generation increases as cells become more mature neuronally. And also we see with FAD mutation an increase in overall tau levels and later phospho-tau levels when we direct these cells to a cortical fate, a vulnerable cell fate, but not when we direct them to another cell fate. We also are able to see effects of A beta from these FAD neurons on GFAP expression in astrocytes. And we believe that directing differentiation to different neuronal fates can alter ABP cleavage. And we're hoping that microengraving in conjunction with some of these other assays I've discussed today will help us better understand the mechanisms by which A beta is generated and hopefully develop new ideas and new targets for how to prevent the toxic form of its generation. I want to thank everyone in my lab, but especially I want to highlight Cristina Muratore, shown here, who is the postdoc who has led the iPS cell effort. And of course, I also want to thank my collaborators, Dennis Selko, Dominic Walsh, have helped a lot with our ability to detect A beta efficiently. Chris Love is our collaborator at MIT who helped with the microengraving assay. Phil Dieger and David Bennett, we're working with them to generate some new iPS cell lines as well as to study certain genes that they've identified through genome-wide analyses. And I want to thank my funding organizations. And I want to thank you all for staying and listening to my work.